Greetings from Boston, everyone. My name is Elena Surilov, and I'm an Associate Director of International Engagements at the Office of Alumni Relations. On behalf of Northeastern University and myself, I hope you and your family are staying safe and healthy during this turbulent time. Welcome to our conversation on COVID-19 vaccinations with our two wonderful experts. Before I proceed to our official program, I would like to go over a few housekeeping items. I recommend to put your view in Zoom on a speaker view. There is a button on the right top corner of your screen to do that. Please keep in mind that the only way to ask your question is to type them in the Q&A section. We will not pay attention to raise your hand option, so please refrain from using it. We have two distinguished researchers on this program, and we will run a Q&A section for each one. So without further ado, I would like to turn the microphone to Peter Kuntis. Peter is our community president for our Greece community, and he's double Husky. He received his MBA from Northeastern in '83, and his Master of Science in Economic Policy and Planning in '86. Peter has been the president of the Greece community for the past 12 years, since its launch almost exact day 12 years ago. I've been honored to know Peter personally and enjoy working with him, as he always surprised me with his power of the personal network by bringing interesting speakers to us. Peter?
Good morning. Thank you, Yelena. Well, good morning, America. Good evening, Europe. Good evening, Australia and Asia. And well, thank you for joining us. Well, and without any further ado, I should like to introduce Professor David Lazzi. And Professor Lazzi is Vice President for Northeastern University Innovation Campus in Burlington, Massachusetts, and also the Vice Provost for Research, Innovation, and Development. He's the chairman of the board at KRI at Northeastern and the former Dean of Engineering. Professor Lazzi is teaching mechanical and industrial engineering, and he is the leader of the university's Department of Defense program in expeditionary cyber, counter UAS, maneuver, and additive manufacturing. Well, Professor Lazzi has worked with the Army, Navy, Air Force, NATO, the National Science Foundation, NASA, and the European Union. He studied at the Stevens Institute of Technology, receiving a bachelor's in nuclear and thermonuclear energy, and he continued with his PhD work at Northwestern University on material science and engineering, before he attending the Institute for Defense Analysis for Defense Science Study Group, and well, concluding with an MBA from Wharton School of Business, where he was a Palmer Scholar. Well, David is a dear friend, and David, I would like to welcome you and thank you for joining us. He has authored more than 130 publications, and his work has been cited in over 6,000 publications. He has received numerous awards, amongst them the U.S. Air Force Meritorious Civilian Service Medal, the Ellis Island Medal of Honor, and many more. So the floor is yours, or I should say the mic is yours. Thank you, David.
The virtual interface is mine. Thank you so much, Peter, and it's great to see you. Thank you very much for this invitation. Yelena, thank you. And Yelena, if I can add one more thing about Peter, in addition to his illustrious and highly accomplished business career, Peter has been a professor in our Demora McKim School of Business. So not only are the alumni of Northeastern benefiting from Peter's great work and his giving of his time, but the students of Northeastern University are benefiting and have benefited greatly from Peter. So Peter, I don't know if being a professor also gives you that triple Husky designation, 2 by degree, 1 by teaching. Almost sounds like something out of Paul Revere. But in any case, thank you very much for this invitation. I'm delighted to be here with my esteemed colleague Achilles Gravanis, who will speak afterwards and cover, I think, a lot of the scientific information and details about COVID and SARS-CoV-2 and vaccinations. So I think that will be a highly informative talk. I thought I would spend my time here giving a brief history of the university and how we responded to SARS-CoV-2. I think it's a story that all of our alumni can be proud of, and you know, the end result was a very successful fall semester against serious challenges. And we, you know, the university has, of course, the business of education. The university has the business of research, and the research program, of course, I'll also mention about, because as Senior Vice Provost for Research, I am in charge of the research across the university. So, so the really powerful story around research, of course, is even before everything shut down, we were already in the fight against COVID, and we have been in the fight against COVID and SARS-CoV-2 since January. So over a year now, we have been engaged in research around this. So I'm going to get into those details. I'll be fairly brief, and then, so hopefully, Peter, we can have plenty of time for questions.
Of course.
So the history of COVID, of course, is, well, luckily I made it back to the university because I was over in Asia, and I arrived back on March 8th, and March 13, everything shut down. So luckily I made it back to the States. I was not stranded, and therefore I was able to be right in the middle of it in terms of working to make the university a success. You know, at the point where we were shutting everything down, there was a big question mark on a number of things. So we were in constant contact with the authorities on how to execute the shutdown of the university, but also working to educate the authorities on how critical it was to keep parts of the university operating. Most importantly, research that was ongoing in terms of COVID, in terms of the SARS-CoV-2 virus, in terms of the spread. So just to give you a sense of the few things that were already going on at the time, we have a network science institute, which is, you know, we would argue the best in the world. We were the first university in the world to create a PhD in network science, and that is, you know, been a very successful program. And we have the lions of the field, both in terms of senior faculty as well as up and coming junior superstars. And back, gosh, maybe several years ago, there was a recognition that disease and the spread of disease in this networked world needed to be described and could not be described mathematically. So if you look at the most famous pandemic of all, which of course was the bubonic plague, Black Death, that swept from Asia across Europe and ended in Scandinavia back in the Middle Ages, that disease traveled by the mechanism of diffusion. That is, nobody had air travel, so everybody was, you know, bumping into their neighbors, villages, trade routes between villages, etc. And therefore, it was always at the intersection of groups where the disease would spread. And therefore, if you chart the records of when Black Death happened, you can actually back out the speed, the diffusion coefficient they call it, but the speed at which Black Death traveled from Asia to Scandinavia, and it was 4 kilometers a day. So 4 kilometers a day was the speed which that pandemic traversed the world, starting over in China and ending in Scandinavia. And or the world at that time, I guess is the way to say it. So, but in the networked world, the mathematics of spread is much more difficult. And there, you know, powerful computers are needed to do that. So several years ago, Northeastern, through its Network Science Institute, started getting involved seriously in trying to develop predictive capacity around the spread of disease in the networked world. And we were called upon, we developed this expertise, we became quite well known for it, and we were called upon by the World Health Organization, the US Center for Disease Control, to help model Ebola. Remember the Ebola breakout that happened that actually spread beyond Africa, as well as Zika. I don't know how much the news about Zika, but it was kind of a big deal over in the Western Hemisphere for a while. So these tools and these models had already been developed. So as soon as a word came of an emergent new thing, and remember that there wasn't a tremendous amount of transparency out of China at the time, early in 2020, but there was informal contacts between scientists from China to the rest of the world. And even without any government officially talking about the presence of SARS-CoV-2, we were already working on it. And because we were working on it very early on, because of our existing relationships, we were immediately pulled into the World Health Organization and the Center for Disease Control. And then when the White House Task Force was spun up, Northeastern was one of the 5 universities around the world that was advisors to the White House Task Force. So it was Northeastern, Harvard University, Columbia University, the Institute for Health Metrics and Evaluation, I think, at University of Washington, and Imperial College in London. So these 5 universities, if you remember back in the early months of COVID, the big debate was around flattening the curve. Do we actually reduce economic activity, get folks isolated in order to flatten the curve? The models predicted that without this social distancing, without wearing face masks, etc., spread would lead to about 2 million deaths in the United States and a lot of deaths worldwide. And the death toll has not been fabulous, but the death toll in the United States, which is a tough place to handle in terms of human behavior, is at 500,000, just past the 500,000 mark. So it's been a pretty significant impact, but nevertheless, a lot better than the predictions were at that time. And also, that flattening the curve gave time for therapies to be developed. So we are doing a lot better now in terms of handling COVID-19 when people get sick than we were at the beginning. Okay, so at the beginning, we wanted to keep this research going. We also had some research that was dependent upon long time period experiments. And if we stopped them, we would lose years and years of work. So we worked with the state to get permission to keep the labs open. Of course, this was all pre-testing. Testing was available, but it was so overwhelmed that it was just not available in a realistic time frame. So this was all done in terms of wearing personal protective equipment. The university aggressively went out very early and very quick. And that's one thing that we're very good at Northeastern, is making a decision and moving quickly. So we were able to procure enough personal protective equipment for our police force, for our researchers, etc. And then we actually got an excess amount. So when the hospitals had trouble in the Boston area, we actually donated a fair amount of protective equipment to the hospitals in the Boston area from the supply that we had at Northeastern. Meanwhile, on the education side, we pivoted over a weekend to online education. That was a tremendous, if you will, turning of a super tanker from classroom instruction, which of course has been the model of instruction, close teacher-student engagement, etc., into one which was 100% online. And that was a big, for some, traumatic in terms of for students as well as for faculty, but people were living the moment of the pandemic so that it was, I mean, I think we can all be very proud about how people responded. People just buckled down and did what they had to do, and then worked to deliver as good as possible an experience, an educational experience for the students. And we know now that we had the summer to seriously tool up, we know that the online education experience in the spring of 2020 certainly wasn't close to the caliber of online components in the fall. Very quickly, gosh, we were still in March, and already at the senior leadership, I sit on the cabinet, on the president's cabinet, at the senior levels of the university, we were already talking about the fall semester and what we could do in the fall semester, and starting to actually game it. So we had a series of task force. One task force was the Research Continuity Task Force. I ran that task force. I also spun up a modeling task force where we applied all these serious modeling capabilities that we used to help the World Health Organization, et cetera. We turned those towards the university and we started applying using students. We brought more students in. We brought faculty and professional researchers in to model the university and how we could keep the university safe, constantly feeding in the new knowledge that we were gaining about SARS-CoV-2 and COVID-19. Remember, this was a real-time exercise back in April and May as new data came in. But in May, we felt confident enough that President Aoun declared to the world that Northeastern would open, and he made the declaration that it was a responsibility of universities to open, and we should open, because at that time he felt that we had the technical capability to actually run a safer environment than the normal community and public environment, especially in the United States. So with that confidence, we have a president that does not play the game small. He went and announced to the world by taking an op-ed in the Washington Post. So in the newspaper that is right in smack dab in Washington, D.C., he announced to the world that Northeastern would open not in spite of COVID-19, but because of COVID-19. And at that time, we had already made the decision. We went and looked at the ways that we could safely open. We recognized that we had to do testing. We looked at all the technologies of testing. I was in charge of that because I was the head of research. So I'm the belly button that the president pokes whenever there's something technical and strategic to work on. So we looked at all the technologies. We looked at all the business routes around these technologies and came to the conclusion that we needed to control our own destiny. So we would spin up our own testing laboratory. And I'm very proud to say that we were able to do that in just a few weeks. We got immediately with our strategic corporate partnerships. And of course, that was an advantage of Northeastern. We already had existing corporate partnerships with, for example, Thermo Fisher, with Agilent. And because we have this strong engagement with industry already, we were able to very quickly tap into those relationships, get meetings very quickly, make decisions very quickly on which technology to use. And we ended up going with Thermo Fisher and their technology for PCR, that's polymerase chain reaction based testing for the virus, looking at 3 locations on the genome. It's the most robust test for SARS-CoV-2 that exists in the marketplace right now. So we put this together very quickly, got the relations together, got the equipment in, and got the lab to a place where it was up and running to final inspection in 6 weeks. Today, to this day, that is the fastest a medical diagnostic laboratory has been spun up and licensed in the history of Massachusetts. With all the great hospitals in Massachusetts, Northeastern holds the record. And when we launched that lab on August 17th to get ready for the fall semester, in one day, it went from 0 samples to being in the top 1% of medical diagnostic laboratories worldwide by volume. So we went from 0 to top 1% in one day. So, so over the summer, in addition to getting ready for the safety, if you come to the campus today, you will see the campus looks incredibly different. Everything is labeled. Every table has labels that show where people can sit in order to have healthy distancing. Every elevator, every staircase, every room in the campus, and our campuses outside in Burlington, Nahant, and Portland, around the country and the world, have labels that show how to do healthy operation of the university in the face of a pandemic. In addition to that, we modeled dining. Dining was our big worry because that's where people have to have their face masks off. We developed marketing campaigns to get the students on board to owning the responsibility. And I tell you, Northeastern students with their experience with industry and out in the real world, they latched onto this, I think, in a more mature way than most universities did. But we paid attention, and this is, people laugh about this, but we paid attention to two very important axes. So Peter, from the business world loves a quad chart, right? I mean, a chart that has, 4 quadrants, right? And you want to be on the top right. You always want to be on the top right. That's where things are good. Well, we had a 2-axis chart where we had these 4 things. The x-axis was safety. We were paying attention to safety. I discovered a new room-sized purification system that used UVC light and HEPA filters that was being used in order to reopen dentist's office where people's mouths have to be exposed. So they were installing these. They were approved by the American Dental Association, et cetera. It was a small Canadian company. So I contacted the company and we went and bought 500 of those units. And we put them periodically in every dining hall just to maximize the purification of air in the riskiest places on campus. But we made moves like that. So safety we took very seriously. The y-axis was joy. And we actually had a joy task force. And the joy task force was, how do we maximize the powerful, positive educational experience that Northeastern has been for many students in the face of a pandemic? And we recognized it would be a challenge. And even with our concentration on this that has led to tents outside and fire pits and special activities where students can do things with healthy distancing, places for students to safely get out of their dorm room and engage in teaching and classes when they are not in the classroom. All of these things came out of our concentration on joy. How to make the testing experience so fast that you could get in the door and out the door with swabbing your nose for COVID and have it be under 5 minutes. So the impact on your day would be negligible in terms of getting tested. So all of these things, and we tried to maximize safety and joy. And that was really the serious way that we looked at, right up to the level of the president. And we presented this to the Board of Trustees, how Northeastern was going to maximize safety and joy. And we have done it, but at the same time, we have paid attention to student mental health. Our reach out to mental health counseling increased 25% from normal in terms of the fall semester. The end result was we ran a very successful fall semester. Yes, we had students that got sick, but we had very quick, testing every 3 days. The reason why we tested every 3 days, as Achilles will attest in terms of the science of COVID-19, you get infected on day 3, you become infectious. So we wanted to be inside the viral transmission cycle. So that's why we chose every 3 days. It was based on modeling by these world-class modelers, and the students have complied. We had some episodes where we kind of emphasized the importance of compliance. Some of them were, and we made big news about it. One of them, of course, it went public and nationwide when we expelled 11 students for illegally partying in a room without masks, and we expelled them for the semester. And boy, that went nationwide, and we had haters and lovers on social media at Northeastern University. But boy, did the students get the message. And then, the students, we set up an organization called the Health Ambassadors, and they were just a powerful. It's amazing how much students listen to fellow students more than they listen to authority figures. For those of us with children, we can't understand how that could be the truth. But in any case, all joking aside, the health ambassadors were a tremendous and powerful thing. So, we went through, we tested every day. Unlike other universities, we tested on Thanksgiving, which is a big holiday in the United States. We tested on Christmas, we tested on New Year's, we tested through Hanukkah, we tested on all of the days of the year, in order to make sure we kept a safe campus. And if you look at the numbers, the infection rate of Northeastern main campus, embedded in Boston, where a lot of our students are constantly interacting with the community, we still kept our positivity rate on campus more than an order of magnitude lower than the Boston positivity rate. So it's been a very successful time. I think, Peter, I will stop there and answer any questions.
Okay.
And then we can get it over to the science and Achilles.
Oh, thank you very much, David. And the first one is, sounds like a very big plan, comprehensive, very successful, sounds like a big investment. Would that be possible to give us some numbers and how much and where did this investment go?
So actually, one of the things I didn't mention was we completely retooled the classrooms. So we could actually run our classrooms both in person and online simultaneously. It's a program called New Flex. And by the way, one of the things we also did was we created a new online experience for the students called the Student Hub. This has been enormously powerful. So Peter, you're in marketing. You're in marketing. So you tell me, if this is a good product, okay? The Student
It's an excellent product.
Well, I'm going to tell you that I'm going to give you data and you tell me based on the data. The Student Hub, on average, every student at the university spends the first 7 minutes of their day looking at Student Hub. Is that a good product?
Wow, it's a great product. Well, they have to focus on this, huh? Right?
It's a great product. And because we made it so that the student could see their entire life on Student Hub, so they could put their social media platforms there, their schedule was there, whether their class was going to be online or in person, their testing schedule was there. Are they due for a test for COVID? What was their appointment for COVID test? Everything was on, their direct link to their email, a one button push to get to their advisor. You want to talk to your advisor? Click this button. Automatically you're open to an appointment window for your advisor. So, you know, it really was some great product. So all of this investment, the official party line of the university is that it was more than 50 million and not yet $100 million investment. So it is a measure of Northeastern, and especially, you know, under the presidency of President Aoun, we have done tremendous innovative things, and it has made the university financially very strong. So for example, we're very proud of the fact that while like other universities, we froze salaries during 2020, other universities, for example, stopped making contributions to retirement plans. So the university matched to the retirement plan, which is a standard benefit in universities in the US. Many universities put a hold on making that contribution. Northeastern did not put a hold on that. So we have been contributing to employees' retirement right through COVID. And we're able to do it because, you know, even with our loss of international students because of travel restrictions, even with that hit to the university is so popular, is running so strongly in terms of worldwide recognition and desire for students to be engaged that that we've been able to, you know, we believe we're going to complete this fiscal year in the black, which is even with the expenses of COVID. Of course, in the fiscal year in the United States for universities runs July 1 through June 30.
And despite the COVID-19 pandemic, with a no record in applications, you know, no students applying to Northeastern University, right?
Unbelievable. We did not think, you know, we had been going higher and higher every year, the most applications of any private university in the United States. We were at 62 million applications for 2019, September 2019. And, you know, and we were worried about what the pandemic would do, but also we always thought that, you know, we could only go up incrementally. 75 million. We jumped from 62 million to 75 million applications during the year.
Let me correct 1,000, but that's okay. Yes, 62,000 versus 75,000. We're all Northeastern.
Thank you very much, Peter. Yeah, 75,000. No problem. Well, that was a little bit of hyperbole there, but what's 3 orders of magnitude among friends?
Right. I can't refuse this.
You're absolutely right. 62,000 to 75,000. That 75 million would be a real achievement. We're going to shoot for that someday.
Okay. Thank you.
May I ask David?
Yes, Achilles, go ahead.
This
Yes,
is
of course.
actually an impressive record, David, in terms of efficacy, and not only that, but responsibility for the students, which is very important, actually. It's an investment, I would say. So Northeastern is an interdisciplinary, field of science. And as you mentioned very well, COVID-19 needs lots of different sciences to be taken care. So you have a privilege of investing early towards this direction. What is the policy for the next years? Because it won't be the only pandemic of our times. I'm sure for that. So what do you plan to invest in the next years by establishing a common and stable actually network of interdisciplinary scientists to study this type of problems, not only in terms of biology or chemical biology or computer science, but also engineering, because Northeastern has all this capacity to have an interdisciplinary research team for COVID-19 and the next pandemic for sure.
Yes. So it's very interesting, Achilles. This is a fabulous question. So when you're dealing with a pandemic, you have everything from, of course, the biochemistry of the agent that you're dealing with. Issues of understanding mutagenicity, mutation rates, and transmission of effectiveness, etc. But also, when you're dealing with something that can become a pandemic, you very quickly learn that you're dealing with numbers and data, big numbers and big data. So, so if you think about the COVID-19 response, we had, of course, the biochemistry aspect, but the biochemistry aspect was more than just, solving the structure of the genome for SARS-CoV-2. It was, exploring what potential therapies could have interaction. And we had researchers running artificial intelligence and machine learning modeling, once we had the data on the structure of SARS-CoV-2 and also its, surface proteins and the full structure of the virus, we had researchers along with other universities and hospital systems running deep, high performance computing algorithms on what therapies, especially the favorite was, of course, to look at every drug that has ever been approved for human use already. So it's already passed, drug regulatory agency approvals to see which of these could be effective against SARS-CoV-2. And there were, some successes there in those identifications. And then, of course, the development of new therapies, et cetera, the vaccination, et cetera, all of that. But data is becoming a huge, important part of this. So Northeastern, we have, we've been very active in this, obviously the network science folks, but also we have been very active in terms of artificial intelligence, machine learning. We have a new institute for experiential artificial intelligence. So that aspect. Then there is, of course, the epidemiological aspect that I talked about. There is our industrial engineers, multiple of our industrial engineers. I think very few people know this. Northeastern is actually very strong in healthcare systems engineering. And we were the ones that developed models for not only the hospital being able to model impact of COVID-19 infections on its ICU units, but also modeling the infection rate, bringing our epidemiologists together with our industrial engineers to work on how to model the impact on the hospital workforce. Because as you have a pandemic, you can have lots of ICU units if you have no trained personnel to run those because they're all getting sick. That is a big, big challenge. The world, and there's lots of examples. I can go on about these types of examples. But also, Northeastern's impact on SARS-CoV-2 and COVID-19 has been recognized around the world. So Cambridge University in the UK, their master is Dame Sally Davies, and she was the person who ran medical research in the United Kingdom. She started what was called the Trinity Challenge, and when she was looking for founding organizations She reached out with all her connections. So founding organizations are the Gates Foundation, the Clinton Foundation, Microsoft, Facebook, Google, GlaxoSmithKline, Swiss Re, Reinsuring. I mean, really a who's who of industry and technology companies and foundations, et cetera. She reached out to Northeastern University. We are a founding member of the Trinity Challenge, and we have very interesting projects that are going forward through that. So for example, our very strong expertise in geographic information systems, GIS, marrying with our epidemiologists to understand better this problem of a networked world and how we can track the flow of people so that we can very quickly get ahead of potential spread in the face of future pandemics. And then, of course, right over to public policy. We know that SARS-CoV-2 and COVID-19, especially in the early days, could have been handled far better in terms of transparency, in terms of public policy. And there's a lot of learning to be done there. And also, you know, a lot of learning in terms of how to mitigate the downside on the economy. And we have our Global Resilience Institute, and our Global Resilience Institute has now many funded programs that are about, Northeastern has developed a toolkit. Now you've got to consider economies are highly interdisciplinary, interdependent networks of things, right? You have your food distribution network, you have your energy network, you have your transportation network, you have your information network. All of these networks, your sanitation network, all of these networks make modern life possible. And our Global Resilience Institute has developed a 187 data point model that very effectively models the economy and gives political leaders actual things to focus on. So we have now gotten big contracts from FEMA, which is the Federal Emergency Management Agency and the Authority in the United States. Of course, they handle disasters, and they have been given the responsibility to get the economy from going down too far, right, to prevent disaster. You know, there's been a lot of dollars put to that type of activity in the US. Northeastern is at the forefront, not only in the New England area, which was the first contract that we won, but now we are in Texas and Louisiana and other places that FEMA is bringing us to apply this expertise. And you can imagine, Achilles, this is data rich with tremendous opportunities for machine learning, artificial intelligence. And we are at the baby stages of being able to do this. It's just starting to be done. And then the other thing I just want to point out is you know what some of our researchers did. So starting in January of 2020, we had a young assistant professor who got together with Oxford University and Shanghai Jiao Tong University, and three folks that knew each other came together and said, wow, this is going to be big, and there's going to be a need for us to have an open and accessible anonymized database on data on sick people who get sick and what, you know, the outcomes there are, and demographic information about their socioeconomic status, you know, everything about, you know, address, where they live, obviously countries, how much they traveled, all of these things. So they created this open database, and it grew to having, I mean, they just publicized it in the community through word of mouth, and it grew to having, you know, tens of thousands and then hundreds of thousands. And then they hit 1 million, and Google got excited, and Google donated 6 months of 12 dedicated Google programmers to convert this into Google's open cloud architecture. And now this database has, I believe it just passed 10 million unique entries from 196 countries or something like that. I mean, this is going to be a database that is open to the world community of researchers to go in and try to learn about COVID-19 in retrospect, but in terms of developing ways to handle pandemics going forward. And then, of course, the hope is that this open data source will become the go-to place for people in Wuhan, China, who start seeing an episode in the future, hopefully allowed by their government, but without retribution, to be able to start uploading cases they see very quickly, so that machine learning tools can look at that and say, okay, this is just normal stuff going on. Ooh, we're starting to see correlations here. This is looking like something that's emerging that is serious. We got to jump on this. If we, even with SARS-CoV-2, 2 weeks faster warning would have made a huge difference. So it really was kind of that, S-curve type of thing that we were on. We were at a tipping point, and the world has suffered because we went through that tipping point before public policy was able to catch up. And so, I think, Achilles, you understand all of this, but you're absolutely right. Tremendous opportunity, and Northeastern is absolutely at the frontier of pushing this going forward.
Thank you.
Thank you.
So, Peter, I think I've taken more than my share of time.
Well, you had a lot to say, and it's
It was
quite
exciting.
impressive.
Really exciting. Yeah.
Never give a professor too much time.
Well, once a professor, always a professor. That's my motto. So
Indeed.
we got to be careful.
When I was Dean of Engineering, I used to also say that. Once you're an engineer, you're always an engineer.
Okay, so I hope I end up as the Dean of Engineering then.
That would be wonderful for Northeastern.
Okay, sounds good. Well, David, thank you very much for your contributions to fighting COVID-19, not only at Northeastern, but all over the world. Quite impressive record. Thank you for your time. Well, good luck with your projects in Washington, D.C., and we'll be in touch.
Absolutely. And thank you to all the alumni, and thank you to Peter, you and Yelena. Thank you very much. But thank you also for all the alumni that are on the line now, staying engaged with Northeastern. We are your home, and we will always be your home. And I hope that this has been worthwhile for you. Have a great day. Have a great evening.
You too. Thank you. Thank you so much. So at this point, I should like to introduce Professor Achilles Gravanis, who is going to be our expert speaker for the night. And well, Professor Gravanis is a professor of pharmacology at the School of Medicine, the University of Crete, and also an affiliated research professor at our own Center of Drug Discovery at Northeastern University. So then Boston, Athens, Crete, so all over the world, a cosmopolitan person and a very well-known professor of pharmacology all over the world. Well, Professor Gravanis is affiliated research professor at the Center of Drug Discovery and has many projects going on there. He also served as a member and chairman of the Hellenic Society of Pharmacology and many other research committees of the European Union. He is also co-founder of biotechnology spin-offs, BioNature and ReNeurocell. He has published more than 150 articles in PubMed. His research focuses on developing synthetic compounds, agonists of neurotrophin receptors with neuroprotective and neurogenic properties and potential applications in therapeutics of neurodegenerative diseases. Well, I will pass the floor to Professor Gravanis, a dear friend and a very frequent speaker on topics and issues that are of interest to the public, the international public and the Greek public as well. Professor Gravanis, the floor is yours.
Peter, thank you so much for this very kind and honorous actually invitation. Helena, I want to thank you for all this, very good preparation. Of course, Professor Lassi for this actually very inspiring introduction to my presentation. I'm going to follow his path and give some examples of how interdisciplinary collaborations in science gave us fast and effective and safe tools to combat the COVID-19 pandemic. Let me share my So, you know, the last 12 months, we were facing with an unprecedented in our times challenge in public health. The last pandemic was evident almost 100 years ago, and it was the influenza epidemic in Europe, coming actually again from China. A huge amount of scientists, almost, 500,000 scientists all over the world collaborated to develop new tools to combat this unfortunate, actually, bad luck we had last year. And they were not only physicians or biologists or chemists, they were coming from all over the scientific, panorama. And we were discussing, collaborating with engineers, computer scientists, modelists, and people working with, networking and artificial intelligence. So the outcome of all this effort, almost 12 months now, is summarized in this slide. First of all, within 10 months, we had developed almost 300 experimental vaccines, 65 of them are in clinical trials in humans, 12 in phase 3, and we have already 7 of them approved for emergency use in humans. And I will give you some of this, some information on these vaccines, approved vaccines later on. On the same time, we were trying to develop therapies. And we had 2 actually ways to focus on, therapeutic ways. The first one was focusing on the virus itself. And I'm talking about antiviral medicines or antiviral monoclonal antibodies. And the second one was focusing on the host, on the patients that have were infected by the virus. And in this case, we were talking about manipulating the immune response and their immune system to try to ease the problems the virus was creating to them. And some of these problems were really almost fatal. So the last 12 months, we have developed almost 2,500 clinical trials with different actually medicines, potential medicines. And one of the helps we got was by repurposing known medicines for other indications. In other words, medicines that we knew already, and they were described for other purposes. And we were trying, using new technologies to find out if they can be of help also in the COVID-19 disease. The last part of trying to find therapeutic targets in the host is summarized in this slide. And you can see that we have plenty of new therapeutic targets that are now under evaluation and research to find ways to block or even diminish the immune response of the virus produces in the host. And now we have some old medicines that are proven to be very helpful, like antibodies against receptors for inflammatory cytokines. And of course, we are developing brand new chemical entities to try to block the cell signaling pathways involved in the inflammatory reaction, inflammatory response of the host after the infection with the virus. It's for sure that in the coming months or years, we will have brand new therapeutic tools to block the effects of the virus in different physiological systems in the host. One of the informations that were very actually valid in the early days of the pandemic is that we were realizing that this coronavirus was not only hitting in the the system of the lungs, but also can interfere with other systems like the blood, the endothelial cells, the nervous system, the kidney, heart, and vessels. So it was a very complicated, actually, medical conditions, and we lost many in the early days of our patients because we didn't know this reality. Now we know, we have a good knowledge of how the virus affects all the systems, and we developed new ways of trying to combat these effects, these deleterious effects. And this is the reason why now in the IC units, we have better ways of saving people, actually, after the infection with the virus. Let me give you a piece of information on this virus. It's an RNA coronavirus. Its genetic material is RNA. It is enclosed in the virion. The virion has lots of different proteins on it and on the surface. One of these proteins is a very, very important protein, is the spike protein. This one is the protein that is a ligand to our cells. Once the virus is entering into our body, it goes in our cells by binding of the spike protein to a receptor on our cells named ACE2, angiotensin converting enzyme 2 receptor. And once this binding is happening, a key and on the surface of the cell, the virus is entering into the cell and hijacks all the machinery and try through our machinery in our cells to reproduce itself, and then get out, exit of the cell, and infect the other cells around, or not only around, but in other systems too, as I mentioned. Another property of the virus is to infect the presenting cells of our immune system. And once this is happening, the presenting cells present the virus, most of the proteins on the virion of the virus in the immune cells of our body. And firstly, they present the virus, the antigens of the virus to the T helper cells, and the T helper cells present the antigens to two types of immune cells. Firstly, the B cells producing antibodies against the virus, and T cytotoxic cells. These cells recognize our cells that are infected by the virus and kills them. So the cytotoxic T cells are very important in diminishing the production within our body of and the replication of the virus. For our efforts to combat the pandemic of coronavirus, we have a very central, actually, mechanism by which we should go through. And this is immunity, human immunity. Human immunity can be achieved by three ways. First of all, by paying the price, by get infected. Once the virus is entered in our body, it induces the natural response of our immune system. And the natural response is going through all this machinery I was explaining before. Of course, we can induce pharmacologically the immune response by vaccination, by inducing in our body antigens of viral origin that can be taken as a challenge because they are not recognized as self bodies of our organism, and the immune system, our immune system, produces T cytotoxic cells and, of course, B cells producing antibodies. And of course, we have another way. If we are not infected by the virus, or if we are not vaccinated, or we are vaccinated ineffectively, then we can give as a drug, as a medicine, the already prepared, exogenously prepared monoclonal antibodies, mixtures of monoclonal antibodies against the viral proteins. And I'll go through all these possibilities in the coming slides. One of the big questions we had, anxious questions actually, at the beginning of the pandemic was if the virus can induce an immune system, a natural immune system. And we know now that this is the case, and this is a very important piece of information, not only because this type of processing will give us a native immunity, but also because we got clues that most probably if we prepare vaccines, these vaccines will be effective enough to mimic what is happening in nature after the infection. So we know now that 8 to 9 months after the infection of a patient, we have B cells producing immunoneutralizing antibodies. And of course, and this is very important, we have CD4 and CD8 T cells, which are cells that can recognize, as I mentioned, the infected cells of our body and kill them. Now we know that the infection by the coronavirus induces a robust, at least for 8 to 9 months, native immune response. Let's go now to the methods we have and we developed. We had at the past and we developed new methods to produce and develop effective and safe vaccines against coronavirus. First of all, we had the conventional methods by using the virus itself. We know how to use inactivated viruses for decades. Actually, most of our vaccines we're using for decades are developed through this process. So we get the virus, we purify it, we inactivate it, so it cannot reproduce itself within our body when it will be entering in our body. And we have different mechanisms and methods to inactivate them, chemical methods or through irradiation. Then we inject this inactivated virus to our body, and the immune system gets the stimulus to build up an immune response. And now we have some of these old-fashioned prepared vaccines already in clinical use, mainly in China. The Sinovac and Sinopharm vaccines are of this type. A second way of producing an immune response through vaccination is not by giving the virus itself, but pieces of the virus, and in most of the cases, purified antigens coming from the virion, proteins, for instance, like the spike protein. And we have now a number of vaccines already developed and in clinical evaluation of this type. One of them is Novavax, comes from the States. It finishes the phase 3 clinical evaluation, and we will have the interim data are very encouraging, and I think that Novavax will have his story completed in the next few weeks and ask for permission for FDA for approval for emergency use. We have also developed new ways of producing effective vaccines, and by using not the virus, not the antigen, the protein of viral origin, but also using the genetic code that codes and produces the viral proteins, the antigenic viral proteins. And now we have already a number of vaccines. The vaccine, for instance, coming from AstraZeneca and Oxford University, the vaccines from Russia, the Gamaleya vaccine, the Johnson & Johnson vaccine, and the CanSino vaccine are of this type. So we get the genetic message coding for the spike protein, and we enter it in a vehicle, in a genetic vehicle. Most of the time is an inactivated virus, which can infect our cells and transfer the genetic code for spike protein in our cells, but the virus cannot reproduce itself and cannot produce harm. So these vaccines are now under clinical evaluation, and 3 of them are already in clinical use. AstraZeneca, Gamaleya, and J&J, and CanSino also will get most probably permission in the coming days. And I'm coming to the new ones, brand new ones, vaccines which use purified nucleic acids coding for the viral proteins. Most of these vaccines use an antigen, the very important spike protein. So these vaccines use purified DNA or purified RNA entering into our cells. Our cells get the genetic code inside their machinery and produce finally in our cells, in our body, the antigen, the viral antigen, purified, a native purified spike protein. And the spike protein induces an immune response, as I already presented, to the presence in our body of spike protein. These vaccines are vaccines already in use worldly. The Mrna and NIH vaccine, the Pfizer-BioNTech vaccine, and another one that will get permission for clinical use in the coming few months, CureVac from Europe. The RNA is enclosed in a nanoparticle, a lipid nanoparticle, to facilitate the entering of our cells. But we are developing also methods to inject in our body the pure DNA without any vehicle. And this is a future way of vaccinating, a pure way of vaccinating in the coming years against not only coronavirus, but also for other microbes or parasites or viruses. Another method that was developed in a small biotech company in California is by using a very, very small amount of RNA, which is enclosed in a lipid nanoparticle, but also with the mRNA for spike protein, we include the coding, the genetic code for an enzyme, replicase, which can take these copies of mRNA in our cells and produce thousands of copies of them. This is very important because we can inject in our bodies a very minimal, small amount of mRNA, and this mRNA will be amplified within our cells and produce a robust, actually, immune response with a very small amount of vaccine. And this, as you may understand, is very important, is critical when we're talking about vaccinating almost 6 to 7 billion people all over the world. So we need the material, a huge amount of material to vaccinate so many people. And if we have this type of biotechnology, genetic engineering tricks, we can use the production of mRNA vaccines for a bigger number of people. Few information on the advantages of the mRNA vaccines. mRNA is a very unstable with a very short half-life molecule. It's a flash molecule. So when it is entering in our body, it prepares to enter in our cells. Then it expresses itself and produces the final protein, the spike protein, the antigenic protein, and disappears very fast. So in few hours, we won't have any of this material injected in our body. The mRNA material will be totally dissolved. To stabilize the mRNA and make it possible to inject it in our body, we proceeded with changing one of its nucleosides. It is the product of 4 nucleosides. So we changed one of the natural nucleosides with a synthetic one and gave a longer half-life to this molecule, so we can include it, embed it into the nanoparticles and inject it. The formulation, the injectable form of this vaccine could not be possible without the nanotechnologists. So nanotechnology was a very important issue in developing these new, actually, mRNA vaccines. the mRNA does not enter into the nucleus. It cannot integrate into our genome, a big discussion for many months. To enter into the genome, mRNA needs to be transcribed into DNA, and our cells do not have the capacity of doing this reverse transcription. So mRNA is a very clean and very safe way of vaccinating. It is immunogenic by itself, so we don't need adjuvant like actions of the vaccine. So with a small amount of mRNA, we can boost actually the immune response and have a better immune response through this platform. It is easy to update for new mutations. This is very important. You may understand how important is this property. We can update a vaccine of mRNA nature within hours. And then once we have prepared new vaccines, including the new variants, the new mutations, we can produce it massively in tons by using biotechnology and genetic engineering techniques. So we can have the new updated vaccines very fast to combat the new variants that are coming evident now in different sites of the world. The mRNA vaccine has 95% efficacy. This is an unexpected result. Most of the vaccines we know for decades do not surpass an efficacy of 70%. So these type of vaccines are extremely effective, in addition to being very, very safe, very pure. And most of the vaccines are effective in the elderly, which is very important, and very important public health issue, because we know already that all the people are suffering for severe disease. So these people should be protected firstly. We don't know yet the exact duration of the immunity, but we have already enough data to predict that most probably the vaccines will be effective enough for at least 10 to 12 months. We don't know for sure if the vaccine can prevent transmission. Once a vaccinated guy gets infected by the virus, if he or she can transmit this virus. We have preliminary findings showing from Israel, actually, and United Kingdom, showing that most probably these vaccines, the new vaccines, can stop the transmission. And this is very important to block actually the transmission in the community in the coming months. We didn't know at the beginning if we can vaccinate pregnant women. Now we have enough data showing that most probably the vaccination of pregnant women is safe enough. And of course, we know now that we can vaccinate people with allergic reactions, severe allergic reactions. And now we are using the vaccination for immunocompromised and allergic patients. Many people were thinking that the mRNA technology is a brand new one. This is not the case. We know the mRNA technology to develop vaccines for at least 20 years. And this slide summarizes some few of the scientific publications dealing with preparing mRNA vaccines, not only for viral infections or for parasites or for microbes, but also for other health conditions like cancer or metabolic diseases. Actually, BioNTech, the company in Germany, a small company in Germany that developed the mRNA vaccine with Pfizer, has in clinical trials a new vaccine against multiple sclerosis, an mRNA vaccine actually. And there are companies now in the States preparing vaccines against very dangerous neoplastic malignant diseases. One of it is already in phase 1-2 for a very dangerous type of breast cancer in women, HER2, ERPR2 negative tumors, and we will know the results in the coming few months. So the mRNA technology of vaccination will shape and change completely the way we will prepare safe and effective vaccines in the coming years. Now a huge actually effort is undertaken worldly to produce within the current year at least 14 billion doses, And most probably by the end of 2021, we will reach this number, so we can be very helpful in many people all over the world. And question that was raised in last year was, okay, most of the time when we are developing a vaccine against a virus or a microbe, we need at least 3 to 10 years to develop a safe and effective vaccine. How come, and in the case of coronavirus disease in COVID-19, we succeeded to have actually dozens of effective vaccines and safe vaccines within 1 year? This was because a huge mobilization, world mobilization was undertaken in the last 10 months, and science, global science prevailed. And academic environments and academic research labs played a central role in this effort. Let me mention again that the Pfizer vaccine was developed by a small academic company. The Mrna Vaccine was developed in close collaboration with NIH. And of course, the AstraZeneca vaccine was done by collaboration with Oxford University. So what was happening the last months is that all these stakeholders, non-profit organizations, foundations, the WHO, the World Bank, collaborated with the industry, the academia, the governments, the states, and of course the regulatory agencies like FDA to fast develop safe and effective vaccines. And they were collaborating in parallel. The production of data were following up by all the stakeholders daily. So we didn't lose time to finish the clinical trial and then evaluate the outcome, these data. We were evaluating every day the data in a huge collaborative study all over the world. And that's how we succeeded to have very effective safe vaccines. This successful collaboration, I think, will change completely the way by which we develop, clinically develop new drugs. And now the associations, the patient associations, because of this success, will push to recopy this type of working together in the future. Few words about the new variants. We know now that 3 of these variants are very important because they are present in huge numbers all over the world. The first, which was localized in United Kingdom, was the B117. The second one comes from South Africa. A third one comes from Brazil. Of course, we have variants coming from other sides of the world. The new ones coming from California, the P1427 and 429. We have very few information about this, but one thing that we already know the last months is that how these mutations change actually the biological behavior of the virus. Most of the mutations are located in the important spike protein and in the N-terminal, actually, of the spike protein. And the mutations open the trimeric spike protein in a way that is better recognized by ACE2 receptors in our cells, and facilitating this way the binding and the entry of these variants into our cells. We don't know yet if these variants are more dangerous in producing more severe pathologies. But this is what is very for sure now is that they have a better infectivity, and that's why we are getting more numbers of these variants in the last few weeks. We are having already a good piece of information on how the vaccines, the already developed vaccines, behave in terms of the new variants. And most of the vaccines are very effective, almost equally effective with the initial variant, the one European variant, almost 80 to 85%. So we are not, we don't have any big problems on the vaccination programs, worldly vaccination programs, using the vaccines like the Pfizer or Moderna or AstraZeneca vaccines in combating these new variants. The efficacy of these vaccines to block the variants from South Africa is lesser, but this does not mean that the vaccines are not effective enough, because you can have an efficacy of 50 or 60% to block these variants, the variant of South African, but most probably this 50 or 60% is adequate enough to effectively actually block the infection. And this is the case for other vaccines. We know this story for other vaccines. For instance, the influenza vaccine has an efficacy of 40 to 55, 60%, and still is able to block the infection in the community. So most probably for these two variants that are prevailing now in the community, our vaccines, the vaccines we have developed, will be effective enough to block the spread of the coronavirus. Another way of acquiring immunity is through pharmacology. And we can prepare now human monoclonal antibodies, a mixture of them, against different antigenic proteins of the virus, of the coronavirus. There are different methods using genetic engineering technologies to massively produce human antibodies against spike protein. Two of them are summarized in this slide. We can get the gene coding for the spike protein into an expression plasmid, into a circular genetic material, infect with this material human cells, HEC293 cells, and produce the antigen, the protein, the antigenic viral protein. This antigen, we can inject it in animals which are transgenic animals and having humanized immune cells. In other words, their immune cells, they are not of their origin, but they resemble and can simulate the immune response of humans. Once these animals immunized, they produce not rat or mouse monoclonal antibodies against the antigenic protein, but purely human antibodies. We can isolate the spleen and hybridomas with myeloma cells of these animals, propagate them in culture, and make them as factories to produce human monoclonal antibodies against the virus. Another mechanism, another method of producing purified human monoclonal antibodies against the virus is by expressing, by isolating and purifying antibodies from our body against the virus, cloning these antibodies and producing cDNAs of these antibodies, express these cDNAs in HEC293 cells, which become factories of a huge amount of purified human monoclonal antibodies. These are the antibodies that are given to the ex-US president with a big efficacy, and there are now at least 3 industries in the US producing effectively producing and massively producing these monoclonal mixtures of monoclonal antibodies. These antibodies are given to mild and moderate infection in the early stage of the infection, and by this way we can immunize these patients and diminish the possibility of becoming severely ill, entering into the hospital and the ICU unit. In addition, we can use these mixtures of monoclonal antibodies, humanized monoclonal antibodies, to protect healthcare workers, and of course to protect groups of the community with high risk of developing and dangerous actually COVID-19 disease. And these antibodies, mixture of antibodies, will complement the immunization of the community achieved by the vaccines. And last but not least, we try to develop small molecules which will be given per dose by mouth in people that are getting infected by the virus in their houses, and trying to block the propagation of the virus in-house, not getting these patients into the hospital. And this new type of developing new small molecules against the viruses were very well actually propagated and accelerated by technologies like in silico modeling and crystallization of the two main enzymes the virus uses to propagate itself within our cells, the polymerase, RNA polymerase, and protease. We know now the crystals of these two enzymes. We have them in silico in computers, and we are using actually artificial intelligence and machine learning to try to screen different chemical libraries, synthetic chemical libraries, natural chemical libraries, to find out ways, keys, to block these enzymes and to develop lead molecules for potential evaluation and development, clinical development of small molecules that will combat the disease in-house. And I will finish with few summary notes. Vaccination may cover 60% of the general population of the world by the end of the current year. We have already almost 10 to 20% of our people in the community already with natural immunity because they were infected symptomatic or asymptomatic by the virus. So by the end of this year, beginning of the next year, most probably we will have the cutoff of 65 to 70% community immunity that will block the virus in the community. Antiviral monoclonal antibodies may be very helpful in limit hospitalization admissions, and by this way is the way our health systems are working with the patients. But we need criteria of in which patients to use this very precious actually material for combating the disease. From next year, we will have most probably the first coronavirus specific perose antiviral small molecules and enzyme inhibitors, which will help us control the infection in the early days at home. And of course, this is very important, as we discussed with Professor Lassi before. All these technologies will help us to effectively and fast act in the new pandemic that will come for sure in the coming years. Thank you very much for listening.
Well, thank you so much, Achilles, for delivering such a wonderful presentation. The timing cannot be more applicable than right now because many people are still trying to figure out whether the vaccination is the right path for them to go. And a huge thank you goes to Peter, as always. Peter, you were just keep amazing me with your network and with the speakers you're bringing. And thank you for convincing David to tune in and to talk about the Northeastern and how we went through the pandemic. Thank you everyone who's still with us and for staying on this program. And we're looking forward to hear more. But before we leave, we wanted to open the floor for questions. If you have any, just hit the Q&A button.
I do have a number of questions from friends all over the world. And in fact, Professor Gravanis, thank you very much for an amazing presentation. I was looking for the right word. Well, I would like to congratulate you not only for the presentation, but also for being proactive. So you have answered a big number of questions before we arrive at the question and session. So the questions we have, this one is from John, Sydney, Australia, Barbara, I can see from London, UK, and Kostas from Athens, Greece. And the question is, what is a better vaccine? What is the best vaccine? Or they say all three of them. Shall I better wait for a monoclonal antibodies or any other drug? What do you advise me to do? So what do you think?
We are very privileged, Peter, to have a number actually of safe and very effective vaccines. So we don't have any choice. the vaccine that is in their disposal should be taken. This is my advice. All of them, Moderna's, Pfizer, J&J, AstraZeneca, are very effective and very safe vaccines. Depending upon where they live, they should go and vaccinate themselves as fast as possible, because the virus, we know now, that is very dangerous. So whatever vaccine they have, they should go and do it. The new technologies that we are having now, and they are the mRNA and cDNA technologies, all these vaccines I've mentioned are of this type of vaccines, mRNA, cDNA vaccines. So all of them are considered very effective and very safe. And I would say they should go ahead with whatsoever of them and get vaccinated. In terms of the monoclonal antibodies, the monoclonal antibodies may be of big help in people that cannot be vaccinated or not effective after get vaccinated. So now we know that in the US and in Europe and in another also, continents like Australia, the monoclonal antibodies got emergency use, permission for emergency use, and they can be prescribed for these people. So we have not only the vaccines, but also the pharmacological vaccination through injection of monoclonal antibodies. And one very important piece of information for this patient is that vaccination, injection of monoclonal antibodies will last for at least, we know now that, 2 to 3 months. So they are not drugs that will be taken for a few days. They are drugs that can be taken for once, bolus, and being effective for a few months. So it's like a type of vaccination. So because we don't have, for the moment, small antiviral molecules, the monoclonal antibodies, the mixture of monoclonal antibodies are very useful and very effective for people that cannot be vaccinated for whatsoever reason.
Okay, thank you. Then we have Robert in San Francisco, California, and his question is, well, he makes a statement. People do need to put the blame for this calamity, for this global catastrophe on someone, a person, a country, a government. So who is to blame for this calamity, that came upon us? So what's your opinion, Achilles?
Yeah, I'm not an expert, Peter, in molecular virology, but I was reading so much all these months, and I will tell you what I think and what I feel as a scientist for 3 decades in pharmacology. We have at least 100,000 viruses out there, and some of them will change their biology and their genome to be permissive in humans. I think what is happened with the coronavirus is this story in China. And China gave the last decades lots of viruses coming from animals and then changing their genome in a way that can infect humans. So most probably this is the case for coronavirus too. But as humans, we can change behavior and lifestyle to minimize this possibility. We should not eat, for instance, raw meat from animals of this case. I mean, we should eliminate of using as food this type of animals. We should use hygiene conditions so to minimize the possibility of transferring an animal virus to humans. And I think that the lesson we got from the coronavirus, actually the first pandemic of our times, will let us know how to change in terms of behavior. And I think this was the case in China. People now start to discussing to stop behaviors that are very dangerous in terms of creating new strains, new variants, viral variants that are dangerous enough. And this could be the case not only in China, but elsewhere, in Africa, in Europe, and in the States. But, in the States, in Europe, we are very careful in terms of hygiene. So the possibility of getting viruses from the animals towards us is very limited. But in other sides of the world, we must be very careful and educate these people not behaving this way.
Great, thank you. And also, John from Boston, Massachusetts, a good friend of mine, says, when is the next pandemic going to hit us? Well, can you predict? Well, I guess a tough one.
I don't know, Peter, but you know, now we are not naive enough as it was the case last year. This huge mobilization of people, human capital, academics, the industry, investments, huge investments. We're talking about tens of millions of dollars only in Northeastern University the last year. So we are preparing ourselves in a scientific and more robust way for the next pandemic. We don't know when it will come, but this time we'll be more prepared. You know, we had our lessons in the last decade with Ebola and MERS viruses. But because the viruses were, the cases were very limited in the community, we didn't pay attention, we didn't invest. Now with the pandemic case, we were preparing ourselves for the next pandemic. And believing, because I strongly believe in human mind and science will prevail again, not only the coronavirus, but we'll prepare ourselves for the next pandemic. And the next pandemic will be much more faster and more efficient to lose less people, less of our people.
Sounds good. Great. Well, I see we have no further questions. So at this point,
I have a question.
You do.
Yes.
Yeah. By all means.
Natalia Kasturi is asking if there are people who will be not allowed to vaccinate due to some medical conditions. Do you know?
Yeah. Helena, at the beginning of the pandemic and the development of the brand new vaccines, we didn't know if we should go into vaccination to specific groups of patients, like the immunocompromised patients, with malignancies under chemotherapy and adenoplastic therapy. Now we know that in most of these cases, and allergic people, we vaccinate them safely. So most of the people, 95% of the people, don't have any contraindication for not get vaccinated. But for people that are having conditions, specific conditions, special conditions, not to get vaccines, we have the monoclonal antibodies. And in the near future, we will have for sure antiviral molecules that can target not the immune system, but the virus itself, the enzymes of the virus. So the title of the virus within our body will be lesser. And by this way, our immune system will take care of it and stop the infection. So for these people, very, very, very few people actually, in the coming months, we can use monoclonal antibodies, and next year, hopefully, small molecules.
Okay, thank you.
Any further questions? Yelena?
I don't have any more questions. I think Achilles did a great job, as you said, Peter, answering most of the questions during his presentation. Achilles, thank you so much for your time, for being with us. And we're looking forward to get more updates on this in the future. And Peter, thank you again for bringing such a wonderful opportunity our way.
Thank you, Yelena, for organizing these and supporting these and making it happen. And of course, thank you, Achilles, for being a part of our family, right? That's a Northeastern family all over the world. Thank you for joining us and sharing your wisdom and experience as a leading scientist. And thank you, thanks your team too, right? Because you are the head of a team working hard, to get new therapies and new drugs coming our way.
It was a real pleasure and honor being with you, Peter. And hopefully we'll meet each other in Boston in May.
Okay, good enough. So at this point, I should like to thank everybody who did attend this wonderful event with Professor David Lazzi and Professor Achilles Gravanis, both of Northeastern University, and Achilles from the University of Crete. And well, our group included alumni, students, parents, worried parents, right? Seeing their children crossing the Atlantic, getting back to Boston. So David answered all the questions and more. Achilles gave us a very positive outlook for the next 12 months. So getting back to a normal life and of course a normal face-to-face education. Well, thank you all once again and have a wonderful evening. Goodbye.
Thank you. Thank you. Goodbye. Thank you.
Thank you. Bye.